Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last couple of classes, we discussed some of the methods that could allow us to do read mapping. Okay. So, we talked about different ways we can do this, and we looked at the drawbacks of those methods. So, in this class we will continue our discussions on two different mapping algorithms. So, this class will give you an overview of the process by which we can do the mapping, right. So, these are the algorithms that drive some of the software tools that we will use in the future classes when you do the read mapping hands on. Okay. So, it is good to know how these methods work, and also we will understand the drawbacks of this method. Okay. So, this is what would be the topic that we will be discussing in this class. It is called hash table based mapping algorithm. We will discuss what is hash table and how this mapping algorithm works, and then we will take some examples, so that you understand how this process actually operates. Okay. So, these are the keywords that will come across. So, seeds, you have seen this before in the last class. We discussed what are seeds that we generate from the reference sequence or from the read sequence, that is the query. We will come across the term hash and we will talk about the term keys. Okay. So, just to give you a brief overview of what we have discussed. So, we talked about the trivial mapping algorithm, which is the brute force approach. So, we use a sliding window to map the read against the reference sequence. But as you can imagine, this is a very slow process, and we calculated some time, right, based on certain assumptions, and it appeared to be very slow, right. So, this is not fit for this purpose. We then talked about the BLAST algorithm, and we talked about the method by which it works. It uses something called seed and extend. So, we generate seed sequences from the read or the query data and search against the reference. And it is still slow because this process requires this linear search in the reference sequence. We talked about an alternative to BLAST, it is called BLAT. So, it is BLAST like alignment tool. It utilizes something called indexing of the reference sequence. So, the seeds are generated from the reference sequence instead of the query or the read, and we found out this is faster compared to BLAST. There are also, we looked at the data from the paper. It was significantly faster than BLAST, but it is still not fast enough for our purpose, and the purpose is to map millions or billions of reads, right, in a short period of time. Okay. So, the search for better algorithms continue, right. So, this is something what we will be looking at, right. So, we need more efficient algorithms. So, by efficient we mean it should be able to process a very large number of reads, and this number is also going up as the technology progresses, right. This is crossing from millions to billions now, right. So, if you are processing multiple samples, you will probably be handling billions of reads. So, the mapping time for these billions of reads should be reasonable, right. So, preferably in hours and not in days, especially not in tens of days, right. Maybe a couple of days, 3 days for billions of reads, that is probably what we can live with, and it should be able to handle mutations and errors in the sequencing data, right. So, this is something that is very important. And it compared to other kind of applications where we have perfect matches, right. So, if you are looking for a pattern in a text or something, we are mostly looking for perfect matches, but here when we are generating sequencing data, we have genuine mutations, right. These are biological variations that we observe in our samples, and also errors in the sequencing data, right. So, we talked about why the errors come and how these are generated for different technology platforms, right. So, these errors should also be taken into consideration, right. So, the algorithm should not be able to perform just matching perfect data, right. It should also be able to get map these imperfections, right. It should be also be able to handle these imperfections. Okay. So, there are 3 types of algorithms broadly that have been developed to do this kind of tasks, and we will actually discuss all of these in brief. Of course, we will get an overview, not into the detailed mathematical discussion or the implementation at the coding level, right, how these are implemented. We will talk about the algorithm, but if you are familiar with programming, you can actually implement these methods yourself. Okay, so the first one is called hash table based mapping algorithm, which will be the topic of discussion today. The next one is called suffix tree based method or suffix array based method. So, there are two alternative ways of doing this, and we will discuss them in the next class. We will talk about the advantages of each of these methods also, and then finally we will talk about the Burrows-Wheeler transform based algorithm. Okay. So, this will be our final algorithm for the mapping. Okay. So, let us begin. We will start with the hash table based mapping algorithm. Okay. So, how does it differ from whatever we have discussed so far, the BLAST and BLOT? Okay. So, the major idea in hash table based mapping algorithm is that we build seeds from both the read and the reference sequence. Okay. So, in compared to BLAST where we build seeds only from the read, right, or compared to BLOT where we read where we build the seeds from the reference sequence, right. Here we build seeds from both the read and the reference sequence, right. So, what are these seeds? You remember, right, these are the substrings of certain length, right. So, we can choose the k value, right, the length of that the length of those substrings of the seeds. Okay. So, once you have built these seeds, right, for the reference sequence we store them in a hash table and along with their locations in the reference. Okay. So, we will take an example to understand how this is done, and I will also describe what is a hash table. Okay. Now, what this hash table does, it actually converts these strings or the substrings to something called keys. Okay. And for each key there will be a value, so that value would be the location. Now, the moment you do this, so this is a data structure, the moment you do this, it allows for very fast search. Okay. You can very quickly look up the hash table and find out the location of that seed. Okay. And once we have stored these reference seeds into the hash table, we can generate seeds from the read and we can search against the hash table, right. So, what we are searching the seeds which are stored as keys, right, and if there is a match in the key, right, we will get a value and that value will give the location of that seed. Okay. So, this is kind of the brief idea, but we will elaborate this further with more examples. Okay. So, there are various options, right. When you are trying to design the algorithm, you can think about different options and how you want to proceed. Okay. So, the first option is seed size equals to read length. So, we say okay, so we choose the seed size as the read length. So, if you have read length of 50 base pair or 100 base pair, that would be our seed size. Okay. So, if that is the case, what we would have to do is we then break down the reference sequence, right, to generate seeds of that size. If it is 100 bases, we generate seeds of length 100, right, and then store them into the hash table. So, if you have read size 100, so there are about 4 to the power 100 possibilities, right. Why is that so? Because for each position you can have either A, T, G or C, right. So, if you consider all possible combinations for these 100 positions, this will be 4 to the power 100. So, it comes out around 1.6 into 10 to the power 60 possibilities, right. So, this is a really huge number. So, of course, in a genome sequence perhaps you will not have so many of them, right. You probably will not get all of these combinations, but at least you will get a large fraction of these, right. So, even if it is 10 to the power 20 or so, that is still a huge number to store, right. So, if you want to store this in the memory in the hash table, right, you will need a huge amount of memory. Okay. So, this is one of the biggest challenges, right. So, you probably will not be implement this in a desktop computer or even a normal server. Probably you will need a connection like a compendium of servers or maybe a supercomputer, right. So, that option is not a feasible option, right. So, what is the feasible option? The option 2 is probably look at seed size that is between 10 to 20 bases. Okay. So, again this can vary depending on the read length that you work with, and researchers have experimented with different read lengths and have seen what works best for whatever read size you are working with. Okay. So, again we extract the seeds or generate seeds from the reference sequence and store in the hash table, and if we have seed size of 10, right. So, this is about 4 to the power 10 possibilities. So, which is close to a million possibilities. Okay. So, this is quite manageable compared to the earlier scenario. And once we have done that, we generate these seeds from the read sequence and then search against this hash table, right. We have million possibilities, and among them we have to match, right, from the read. Okay, so let us look at how it works, right, and we will take some examples, right. So, let us take this example where we have this reference sequence. This is an arbitrary example, and we are working with small reference sequence and read, so that it is manageable, right. We can complete this example here within this slide and within a reasonable amount of time. Okay. The process actually is the same, it is just will take more time, more memory, and of course, more elaborate. Right. So, here is the reference, and then you have the read sequence, which is the query, okay, and seed size we choose as 5. Okay. You can choose any other seed size, does not really matter. Okay. So, here are the seeds that we have extracted from the reference sequence along with their positions. Okay. So, if you look carefully, right, what we are doing, the size is 5, right. So, we look at first 5, right. We take first 5 letters from the reference, okay, and we build this seed, okay, and this is the first key, okay, and here we have the location. So, the location is 1, this is the first position where the seed is present. So, we add this in the value, okay. Then we move on the next one, right, GCATC, right, and we add that substring to the hash table. We have the position location, which is 2, okay, and then we go on, right, we slowly move on along this reference sequence, and finally we see this one here GGCAT, and this one was already present, right. So, what we do is we just add the value, add an additional value for that key, right. So, what it means, this key, this seed is present in 2 positions, right, it is repeated in the reference sequence. Okay. So, we need to store these alternative locations, and then finally, we have the final one here GCATA, which gives us this here, right. So, this is the hash table that we build as we go along the reference sequence. Okay. Now, we will utilize this hash table now to actually map our read, okay, the read that we have taken. For the read, okay, so here is the hash table, right, as I have mentioned, these are the keys I have mentioned, and these are the values. Okay. So, these keys, if we can find these keys, right, we can get the values, we can retrieve the values by just calling the keys. Okay. So, this is a specific data structure which we call as hash, and because we have many of them, we call them as hash table. Okay. So, now how do we search a read location, right, using this hash table we have built. Now, once we have built this for a specific reference genome, you can store this in a file, right. You do not have to rebuild this hash table again and again for the same genome, right. So, let us say we have built the hash table for human genome, human reference genome, and we can store this in a file, right. What are the locations, what are the keys that we have for a specific seed size. Okay. But if the seed size changes, then of course, you will have to rebuild the hash table. Okay. Now, this read that we have, now we actually have to generate these seeds again from the read sequence. Now, again we have 2 options. We can say we generate a single seed and try to find that out in the hash table, right, and find its location, or we can go with multiple seeds. Okay. So, let us look at what actually would happen, what are the advantages or drawbacks of each of these, right. So, if we are looking up a single seed, we have generated the single seed, again the k is 5, right, seed length. So, CAGA, we have generated this seed, it is here, right, single seed, and we then look for this seed in the hash table, and here it is, right. We have this CAGA, and we get the value, right. So, which is 6. Okay. So, this is in the 6th position of the reference sequence, we have this seed present. So, what it means is that this read starts at the 6th position, right. So, that information is now available to us after we have managed to search this seed in the hash table. Now, once we have done that, we can simply extend and align, right. We can use an alignment algorithm to see what are the matches, mismatches, etc., and get the final results. Okay. So, that sounds quite easy, and this is really straightforward, right, and it is also it does not take too much time, right. So, you just search for the single one. It is an incredibly fast process if searching through an hash table. Now, what would be the major drawback? Why don't we use this most of the time is because if you have a mutation or sequencing error in the read, this may lead to no match. Okay. So, you may not be able to find that seed in the hash table. So, imagine this example, right. Instead of this A here, you have a T, right. In red, this is the mutation that we have in the read or sequencing error in the read, and when we compare against the hash table of the reference sequence, we do not see any match. Okay. So, if you do not see any match, right, we cannot look at the read, we can say okay, that we do not find the read in the reference genome and we discard it, right. But that would be wrong, right. Just because of a single mutation, if you discard that read, you cannot map that read, that means you are losing data. Okay. So, this is something that we want to avoid. So, probably the better way of doing this would be actually going for multiple seeds. There is another disadvantage, right. So, if a single seed it matches to multiple positions in the genome, right, we have seen it can, right. If you have, for example, this seed here, we can match to 1 and 14, right, 2 positions. You have to now see align this for at both of these positions, right. You have to do this extend and align for both these positions and see which one actually aligns better, right. Again, the alignment part is a time consuming process, and if you have to do this for more than 2 or 5 or 7, right, that will actually take up time. Now, what you can do is okay, so here is this, right. So, again, if you imagine instead of 6 you have 16 24, right, for 3 positions. Here you have this match for this seed, then you have to do this extend process extend and align process for all 3 positions. Okay. So, this takes time, right. So, probably this is better to work with multiple seeds. Okay. So, what do we mean by multiple seeds is that again for the same seed length we have this CAGA, right. We are generating all possible seeds here, right. We are just sliding this window again and we get these 5 seeds. Okay. Now, what we will do now is we will search each of these seeds for their positions in the hash table. Okay. And let us do that, right. And what we see is that this seed actually matches with this one here, right. This seed here, the second one matches with this one here, right. But the rest of them they do not match, there is no match, and I have highlighted this C base here because this is a mutation. Okay. This is a change that is in the read, it could be a mutation or a sequencing error, right. So, these 3 reads are not matching, but these the first 2 have matched. Okay. And the first one is matching, let us say, to these 3 positions. Okay. So, there are 3 values for this first match. Now, in the earlier method you would have to extend for all 3, right, but in this method we can see the first one matches at position 6, the next one matches to a single position 7. Okay. So, this is closest, right, this is close by and there is no other option for the second match, right. So, what it means is that the full read, right, it comes from probably position 6, 7, it covers that position, right, and not 16, 24, right. So, if you have these multiple matches and in nearby locations, you can probably be sure more or less is that this is where the read will map. Okay. And you can go ahead with probably that only one alignment. Okay. So, having that multiple seeds, we can actually do this much faster because we do not have to extend for those multiple positions. Okay. All right. So, it is a process that we call chain align, right. So, we are kind of chaining these locations, right, these they are mapping to, the seeds are mapping to neighborhood locations, and you can simply do chaining and alignment. Okay. So, this is the these are the 2 steps that we have for multi-seed hash table based mapping. It is a 2-step process, it is seed and search, and then you have chain and align. Okay. So, let us look at another exercise, right. Let us look at this, take this example and let us illustrate, right, what we will, how we will find this read position in the reference genome, right. Also the seed size is given, k equals to 3, right. So, let us build those seeds from the reference stored in the hash table, and then we will also generate the seeds from the read, and we will try to find them in the reference hash table. Okay. So, let us do that for reference. Okay. So, for reference 1 we have k equals to 3. So, what we will do is we will generate this ATG, right, its location is 1, we have TGC location is 2, we have GCG location 3, CGA location 4, GSC location 5, we have SG location 6, CGG location 7, GGA location 8. Okay, so we need to just check whether they are kind of repeated seeds, right. So we do not see any repeats, right. If we see repeats, then we have to merge the locations, right. The values will be one after another, right. So we then separate it by a comma. Okay. So once we have generated the seeds now, right, from the reference, we have to generate the seeds from the read itself. Okay. So for the read one, right, we have same seed size, right, CGA, GAC and ACT, right. We have generated these 3 and we can now search the hash table, right. So this is the hash table here, we have the keys and the values and let us search. Okay. So the first one will search CGA, okay, and we see is here, right, and the value is 4, right. So it is matching to 4th position. What about then the next one, which is the GSC, right. So it seems GSC is here, right, 5. What about ACT? Can you find ACT anywhere? Right, I do not find ACT in the hash table, right. So we cannot find this one. Okay. But out of these 3 seeds from the read, we have located 2 of them, right, and they are located at position 4 and 5 in the reference sequence. Okay. So it kind of gives us an idea that probably the read will start from position 4 or so. It's in the region of this position, right, around this region. Okay, 4, 5. Okay. So and as you can see, it's actually in the reference. If we go back, it's actually matching here, right. So this is where it should be mapping, right. So if you write CGA CT, okay, and this is the reason why we did not match, or we could not get the seed in the hash table, this seed ACT, right, because there is a mutation, right, G to T mutation, but the other seeds actually worked well, right. They could give us the location of the seed, right. So in this simple example, I hope it's clear, right, how would you generate this hash table, right, for the reference sequence, and then how do you generate the seeds for the read, and then how do you map these seeds to the reference hash table, and in that process you will identify the location of the read. Okay. So this search process is very fast, right. So you remember, right, in the last class when we were talking about BLAST and BLAT, right, one of the problems, one of the drawbacks we had is that we are doing a lot of comparisons, right. Most of them were unsuccessful comparisons, right, and one of the discussions we had is that we want to minimize this number of unsuccessful comparisons, right. So here you see, like, we are kind of trying to maximize these successful comparisons, right. We are not going to do a lot of these unsuccessful comparisons. Okay. So I hope this is clear how it actually works, right. So there are a lot of tools for that utilizes this hash table, right, hash table based mapping tools. Here is a list of them, right, and you can of course look up, search and see how this works. So you might ask, why do we have so many different tools if they are based on the same algorithm? Of course, they are using slightly different implementations and how they actually go about implementing the algorithm, right. So you can of course have variations in different in the implementation process, and probably you can also add a few bit of optimizations here and there, right. So this is why you have so many of these algorithms. Okay. They combine different ways of doing the same thing, but they are based on the same algorithm that we have just described. Okay. So what are the advantages of hash table based mapping? Okay. So one of the first thing is that it's very fast, right, and if you just go and read literature for some of these tools, you will see you can map millions of reads per hour, Okay. So, that is a very good number. Okay. If you can, let us say, even map 1 million read per hour, right. So, that is within few hours you can map few millions, right. So, this is a very fast algorithm compared to whatever we have discussed, right, BLAST and BLAST. Okay. So, this is very important for us, and it can handle or map reads with mutations and sequencing errors, as we have just described, right. We have seen that even if you have mutations or some sequencing errors, they generate mismatches with the reference sequence. This process can easily handle those, right, because we are working with a large number of seeds from the read, right. Maybe some of them will contain mutations, right, but rest of them will match perfectly to the reference sequence or the hash table, right, entries in the hash table. Okay. So, that kind of allows for this mapping process, right. What are the drawbacks then? If this is, I mean, we have seen this is very fast and we can handle reads with mismatches. What are the drawbacks? So, as you can probably imagine, right, storing that data, storing the hash table in the memory, it will take a lot of space. Okay. So, if you want to store this in a hard drive, also you need to give space, right. So, or during running the algorithms, right, running the tools, you will have to load that data in the memory, right. That is why the whole process is very fast, because this whole hash table is in the memory of the system, and you can simply search these seeds from the read against this hash table. Okay. So, if you have to load this whole thing in the memory, it will take a lot of space. Okay. So, for example, here if you are working with a large genome like human genome, it can take up to 12 to 15 GB of memory, right. So, it means you are not going to run this in your desktop computer, right. So, that is a limitation, means it prevents accessibility, right. So, if you do not have an access to like a, let us say, a workstation with a large amount of memory or a server where you have a lot of memory, you will not be able to run this. Okay. So, this requires specialized systems. Okay. The other process, which is the seed building from the reference or the hash table that we generate, okay, that actually takes quite a bit of time, right, because as you can imagine, you have to go through this window, right, sliding window approach. You have to go through the whole genome, right, to generate this hash table. And if your seed length varies, right, the k that we have discussed here, if that varies, then you have to build this every time you are working with some data, right. So, again the question is, can we not keep the seed length same, right, can we not say, let us say, set k equals to 10 or k equals to 5. Now, this actually is a problem, right. We cannot keep this fixed, because if we are working with, let us say, different data from different platforms, right, they come with a different read length, right, and also they come with different sequencing errors. Okay. So, if you think about this carefully, right, if your sequencing error actually increases, which is the case for long read sequencing, you have to design seeds to be shorter, okay, so that a single seed, most of the seeds will have some match against the hash table, and they will not contain the mutation or the sequencing error, right. So, this is something that will kind of impact your choice of seed length. Okay. Now, the other problem we have is that if you have reads that are mapping to repeats, right, you probably will have some increase in time, right, because if you see there is like a lot of matches for 2 regions, you have to again then extend, align and make that decision, right, whether these read is mapping to multiple regions or not, okay, whether its read is coming from repeat sequences or not. Now, this is a problem that we have just mentioned, right. You have to design smaller seeds, right, you have to reduce k as the sequencing error increases, right. So, so as to avoid that some seeds will contain a perfect match with the keys in the hash table. Now, the moment you do that, what will happen is that you will get too many hits, right. You can imagine, right, if you take a 3-mer, right, versus say 7-mer, you will see more hits for 3-mer in the genome compared to the 7-mer, right. So, as you increase the length of k, you tend to pick up more unique sequences or unique combinations, right. So, if you reduce the length, you will get too many hits, and again we have the same problem, right. We are getting too many hits, means you have to again check probably through this alignment against multiple regions. Okay. So, this will again increase time for the whole algorithm. So, what are the alternative strategies that we have? So, this is an alternative strategy where you have something called seed, filter and align, right. So, we have this seed, right, and this is implemented by a tool like GSST, right. So, what we have is a single seed match, right. So, this tool actually takes a single seed, it matches against the reference hash table, right. Now, let us say it finds multiple matches, right, multiple locations, multiple values. Okay. Now, instead of trying to extend or aligning against all these matches, all these locations, what it will do, it will quickly search, right, this neighboring region for match. Okay. So, for match with other seeds. Okay. How does it actually quickly check? Do you think it is just align or extend method? It is not actually, it is a very fast algorithm based on distance. Okay. So, it is just counting number of bases in the seed versus the neighborhood and see which is likely to be a match, which is not likely to be a match, right. So, if it is a match, you are likely to have similar number of bases, right, in the neighborhood of that seed. Okay. So, it is not using an exact alignment method such as dynamic programming, which is time consuming, but it is a very fast distance based method, right. So, it works pretty well. Okay. So, there are other ways you can reduce now this storage requirement or memory requirement. It is called gap seeds. You can, instead of generating all seeds from the reference, you can say I will store only seeds at certain distances, right, maybe non overlapping seeds, right. So, that will reduce the number of seeds and that will require less memory, right. You can also encode the bases in some ways that you actually require less storage or less memory, right. This optimization can be done without going into too much details of that, and you can of course parallelize the whole process in case you have these multiple matches of a seed in the reference sequence. Okay. So, after this discussion, what do we need actually? So, we need a really fast mapping, which this tool actually does. Mapping to repeat sequences, it should be able to handle these multiple matches very well, which there is some problem with this method. Ability to handle errors. So, this method actually does that very well. Low memory requirement, right, again this is where this method would fail, right, because it requires a lot of memory or storage. Okay. So, these are the references that we have covered, and to conclude, so these hash table based mapping algorithms are really fast and they can map reads containing sequencing errors. So, there is no issue with mismatches, but they require large memory, right. So, this means you have to have special machines, special servers for doing the mapping, and it also becomes inefficient for its mapping to multiple positions or repeat sequences, right. And also increase in sequencing error, which long read sequencing platforms have, right. You see increase in sequencing error, it will require lowering of seed size, which will mean multiple hits and it will increase mapping time. That is it. Thank you.